Image Labgrown colorectal cancer organoid at 10 days. View more. Credit UC San Diego. Health cancers driven by mutations in the CRAS gene are among the most deadly. For decades, researchers have tried unsuccessfully to directly target mutant CRAS proteins as a means to treat tumors. Instead of targeting mutant CRAS itself, researchers at University of California San Diego School of Medicine are now looking for other genes or molecules that, when inhibited, kill cancer cells only when CRAS is also mutated. The team used the CRISPR-Cas9 gene editing technique to systematically inactivate every gene in the genome of human colorectal cancer cells with and without mutant CRAS. They found that growth of CRAS mutant colorectal cancer cells in mice was reduced by approximately 50% when two genes that encode metabolic enzymes NADK and KHK were also inactivated. The study, published September 27 in Cancer Research, provides potential new drug targets for CRAS-driven cancers. We did not get these same results with cancer cells grown in the lab. The growth inhibition we saw when the NADK and KHK genes were inactivated only occurs in tumors in a mammalian system, in a more realistic microenvironment, where the tumor has to survive, said senior author Tarek Rana, Ph.D., professor of pediatrics at UC San Diego School of Medicine and Morse Cancer Center. That suggests that the metabolic dependencies of tumor cells growing in a laboratory dish may differ dramatically compared to the same cells growing in a living system, underscoring potential limitations of standard laboratory-based cancer cell growth tests. Approximately 20 to 30% of all human cancers have mutations in the CRAS gene. CRAS mutations occur in many of the most lethal and most difficult to treat cancers, including lung, pancreatic and colorectal cancer. CRAS mutant cancer cells are able to rewire their metabolism in a way that gives them a growth advantage compared to normal cells. Rana's approach to treating CRAS-driven cancers inhibiting other genes or molecules in addition to CRAS is called synthetic lethality because the intervention is only lethal to the mutated cells. In a previous study, Rana's team used a library of microRNAs, small pieces of genetic material, to systematically block protein production and look for those inhibitions that are synthetic lethal in combination with CRAS mutations. In their latest study, Rana's team used CRISPR-Cas9 to systematically inactivate genes in two human colorectal cancer cell lines, one with normal CRAS and one with a mutant CRAS. They then tested the ability of each of these cell lines to grow as tumors in mice. They found that inactivating two metabolic enzymes, NADK and KHK, reduced the growth of CRAS mutant tumors by approximately 50%, but had no effect on normal CRAS tumors. They also blocked these same enzymes with commercially available small molecule inhibitors and saw a significant reduction in tumor growth in mice only in tumor cells with mutant CRAS. Rana and team also identified several new genes that, when inactivated, had the opposite effect. They increased CRAS mutant tumor growth, but not the growth of normal CRAS tumors. These types of genes are known as tumor suppressors because they normally keep cancer cell growth in check. One of the most surprising findings from our study is how performing this type of genetic screen directly in a mammalian microenvironment revealed not only new synthetic lethal interactions, but also new tumor suppressor genes that are dependent on CRAS mutations, said first author Edwin Yao, MD, PhD, a hematology oncology and cancer therapeutics training program fellow in Rana's lab. One of these new tumor suppressor genes encodes INO80C, a large multi-subunit protein that, among other things, stabilizes the genome. Rana, Yao, and colleagues are now taking steps to carry their findings forward, with the ultimate goal of better understanding how CRAS mutant cancers develop and translating these insights into developing new therapies to stop them. Co-authors of this study also include Indrasana Kametha, Chinlugi Lachinchi, Rachel Tang, and Yanlin Zhang, all of UC San Diego. Disclaimer: Oz and UREC are not responsible for the accuracy of news releases posted to UREC by contributing institutions, or for the use of any information through the UREC system.